Welcome everybody to the UCD's Institute of Food and Health Research Bites. So I'm delighted this morning to welcome Dr. Ronald Halim, and he's an assistant professor at the School of Biosystems and Food Engineering. And his research is in the area of waste remediation and sustainable production of biomass-based food and fuel applications. So his PhD was in chemical engineering in microalgae lipid recovery, and that was done at Monash University. And since then, he's been taking a trip around the world between the University of Melbourne, Karlsruhe Institute of Technology, Carnegie Institution of Science at Stanford University, before arriving here in Dublin last year. So you're well traveled for someone who's early stage career, Ronald. So he has been successful in attracting his research funding in a number of competitive schemes, such as the John Stocker Postdoctoral Fellowship and the Humboldt Postdoctoral Fellowship. So currently his research at UCD is looking to build the capacity for research continuing in the area of microalgal biotechnology and waste remediation. So today, I think he's going to give us a snapshot of his research. So over to you, Ronald. We look forward to your talk.
No worries. Thank you for that, Dolores. Much appreciated. And good morning, everyone. So I guess I'll just get started on my presentation. And yeah. So I guess the way that I structure this presentation is just sort of giving everyone an overview of the microalgae biotechnology field. Of course, it's almost impossible to capture it or encapsulate everything within 15 minutes. So there's bound to be generalization and simplification, but I hope that everyone gets an idea of what the field is about. And then I then kind of move into my specific area of interest within the field and where I see myself in the field and how I will advance it. So, so the presentation today will be about microalgae, the next frontier in waste remediation and bio-based products. And so I've sort of given an illustration here of, I guess, microalgae research being scaled up from a bench scale to a lab scale and then to a pilot and commercial scale. And a little bit of background about myself. So I had my bachelor's degree in chemical engineering from UNSW in Australia and my PhD in microalgae biotechnology from Monash in Australia. And if migration is were a profession, then I will be a profession. I will have achieved my qualification there. So just a bit of background. I was born in Indonesia, and then I moved to Australia many years ago, and did all my qualification there. And after completing all my degrees, I did a number of postdocs within Australia, and then in the US, and then in Germany, before finally landing here in Ireland. And I've started my group since at the end of last year in 2019, and I have enjoyed being here. And thank you to all the supportive. Thank you to UCD for being so supportive. And thank you also to my school, the Biosystems and Food Engineering, for all their support. Now, microalgae. So what is it? Well, the first question, what is microalgae, is probably actually the most difficult one to answer, because it's essentially just a broad-based coalition of single-celled aquatic microorganisms. So any microorganisms that live in water and can photosynthesize are generally considered as microalgae, but you're really covering a very broad spectrum of biological classification there. Now, there's significant biodiversity. So there is, as currently it's up to 250,000 species are known, but it's estimated that there might be up to 2 or 3 million species actually around the, I guess around the globe, though that might be an underestimation as well. Now, there's been significant interest in microalgae over the past, I would say, decade or 15 years, and primarily because of their, because people view them as a promising source for bioactive products as well as biofuels. And the reason for this is, first, they can grow in brackish or seawater, depending on the strains, and they do not need arable land. And they also have very high photosynthetic efficiency. So that's estimated at about between 2 until 20%, as compared to terrestrial plants that have photosynthetic efficiency at around 2%. And as a consequence of this, what it means is that when comparing it to, say, traditional terrestrial plants, microalgae will likely have a much higher aerial productivity compared to plants. And at the same time, they don't actually need as much resources in terms of agricultural resources. So you're not taking resources away from food production or other types of critical food production. And microalgae can, I guess, key things that's important is that I will repeat a little bit later, is that it can assimilate both inorganic carbon as well as organic carbon. So inorganic carbon, essentially the carbon dioxide from the air, and organic carbon, essentially dissolve organic carbon in some sort of waste water or in some sort of aqueous solution. And it also requires nutrients such as nitrogen and phosphorus, just like any other organisms, to synthesize protein and hence for its cell division. So I've put there a number of I guess pretty pictures for the microalgae. So different strains are known for different bioproduct accumulation. Like Nanochloropsis is known to be rich in omega-3 lipids, so that's in EPA. And then I think that's actually a typo there. This one should actually be Pheodactylum, and I just haven't fixed the legend there. But this is Haematococcus, so the third one here. And this one is particularly interesting because as the cell started off when it's young as a green cell, and then as it enters maturity, it starts accumulating the bioactive pigment astaxanthin, and you can notice an actual color change in the cells itself. And so, as I mentioned before, microalgae, they're able to assimilate both the inorganic carbon and organic carbon, and this opens up the gate for the use of microalgae in both waste remediation and carbon capture. So the inorganic carbon, or the gaseous carbon dioxide, essentially, microalgae can absorb that or can assimilate it into its biomass through the standard photosynthetic pathways. Whereas the organic carbon, it actually has the ability to transport or import organic carbon, normally in the form of acetate, glucose, and glycogen, and glycerol. And it imports this through its tricarboxylic acid cycle and assimilate it as part of its biomass. And what this means is that if you have a production system or a process that emits carbon dioxide and generates nutrient-rich waste, in particular organic carbon-rich waste, you can use microalgae as a green and sustainable means to actually remediate all your waste products, or a number of your waste products. So, as I mentioned before, hundreds of thousands of microalgae species, but generally speaking, the field kind of focused to about 15 or 20 well-known strains. And I've sort of listed these strains on this slide here. And as you will notice, the products that we generally sort of focus on are either something related to carotenoids or something related to fatty acids, as well as something related to the phycobilins and polysaccharides, though there is an ongoing research to try to produce a biodegradable plastic from microalgae as well. And the idea is that we can use this microalgae to grow the microalgae and then process the microalgae for the production of all of these bioactive products, which can then be converted for applications in animal feed, fish feed, aquaculture, nutrient fortification for food product, cosmetics, as well as nutraceuticals. Though of course there is a bit of regulatory hurdles to actually be, overcome before these products can be commercialized. One thing that I guess a particularly critical product, or the one that the field sort of really focus on right now, is the production of EPA and DHA from microalgae. And that's critical in nature because the idea is to use the omega-3 from microalgae to replace or to displace omega-3 from fish oil. And global warming coupled with overfishing have made, I guess, the supply of fish oil unreliable, and an alternative source is, I guess, badly needed from a sustainability perspective. And so, as I mentioned before, there is a strong synergy between the agri-food industry and then the microalgae bioindustry. And microalgae can essentially be used as a green and biological or sustainable means to valorize agri-food waste. So it can be used to recover carbon-rich and nutrient-rich waste products from the bio industry or the processing that we are trying to, I guess, attach or synergize ourselves with. And it can also be used potentially to capture carbon dioxide from that process, produce biomass, at the same time generating oxygen. And then this biomass can then be dewatered, releasing treated waste product that's safe for disposal, and then further refined to then produce the specific bioactive products that we desire. Some of the bioprocessing industries, some of the key bioprocessing industries from Irish perspective would be dairy processing and brewery, the brewery industry. I actually have a research project going on with regard to the dairy processing, but I won't actually go too much into detail in this presentation today. And from dairy processing, for example, you actually generate waste streams that are very rich in carbon, such as whey permeate that's rich in lactose, that the microalgae can potentially assimilate and use for its own growth. Now, one of the projects that I guess, is currently under consideration, but perfectly illustrates what I described just now, is the biogas production. So in a biogas plant, you essentially have agriculture and food waste coming in, and then they process it to generate biogas, which will later then be converted to electricity. And microalgae can be used to capture both the carbon dioxide and then recover the carbon, nitrogen, and phosphorus from its digestate as well. Now, with the project that we are pitching, what happened is that we're trying to develop a novel carbon dioxide delivery system to the microalgae, which I will talk about in the next slide. And the idea is to actually focus on a cultivation of native microalgae strains that's adapted to growth in cold water found in Ireland and the UK. So a lot of the strains, a lot of the industrially promising strains were first isolated in more tropical regions. So that's in Spain or in Israel. And as a consequence of that, they may not be very well adapted to growth in colder climates. And they can, but the issue is that you have to then provide heat to warm to increase the temperature of the cultivation system to the required, say, to around 20 degrees Celsius. That by itself is a huge operating cost. So if we can grow something that's adapted to the cold water, that will certainly be much more scalable for Northern European environment. And then the idea is then to use the biomass that we generate from this project as an animal feed supplement to see if that will make a difference to the growth performance of pigs in its post-weaning stage. And the beauty of the synergy between biogas production and microalgae is that biogas producers tend to be located in the countryside, so we are stimulating bioeconomy in rural areas. But more importantly, they tend to actually be co-located next to an animal farm. And if we use the biomass to actually feed or supplement the feed for animals, they then generate the slurries that's needed to supply the biogas plant with its feedstock. So we are demonstrating potentially a circular bioeconomy within a single site. So I've mentioned before that we're trying to develop I guess a new system for carbon dioxide delivery. So the majority of carbon dioxide right now is delivered to microalgae cultivation through sparging. So that's essentially compressing the gas and then pushing it through pipes and then pushing it through the nozzles into the microalgae culture. And the issue with sparging is that the gas simply just doesn't stay in the culture long enough for the microalgae cells to assimilate the carbon dioxide. So what happens is that with a lot of large-scale microalgae cultivation, the carbon dioxide that they pump or supposedly capture from their power plant essentially just gets re-emitted to the atmosphere from the microalgae because it just passes through the microalgae with very little actual utilization efficiency. So our idea, well, I wouldn't say our idea, so there are a couple of groups working on this as well. And for this project, I'll hopefully, if it's funded, I'll be working with Professor Casey and Dr. Stafford Viggers from UCD. And the idea is that we will develop a non-porous membrane for the delivery of molecular carbon dioxide in aqueous form into the microalgae culture. So the ability, the membrane will transform the carbon dioxide into bicarbonate and carbonate ions in the aqueous solution, which will then have the residence time needed for assimilation in the microalgae culture. So I've kind of given an overview of the field there and sort of my interest in the upstream area in relation to dairy processing and biogas production. But now I'm kind of I will sort of go down the value chain of of microalgae production system and talk about the bottleneck at the opposite end, which is the biorefinery system as well. That's the recovery and the extraction of products from the microalgae cells. Now, microalgae products are intracellular and they need to be liberated from their cell walls for their recovery. The cell walls, before I go into the cell walls, so generally speaking, the biorefinery is split into 3 separate steps. This by itself is an oversimplification. The first step is a cell disruption, which breaks the cells open. And then the second step is some sort of a extraction process, which recovers the product from the cell into a solvent. And then the next step will then be the fractionation of that recovered product, as well as the removal of solvent and the conversion of that product to its final form. Now, microalgae cell walls, one of the key bottlenecks in the system is the cell wall itself. The cell walls are composed of tough biopolymers, and they're generally made from a mixture of polysaccharide and glycoprotein complex, which are not easy to disrupt. So they limit extraction efficiency. Either you're not getting enough product from your microalgae cells, or you're consuming too much energy and too much solvent in order to actually recover all the products that you need from the microalgae cells. Either way, you need to, it is important for us to understand how microalgae cell walls work and function in order to improve the downstream processing. So an example I've taken here is nanochloropsis. Nanochloropsis is one of the, well, I guess quite a popular choice for microalgae research and production system, primarily because it's rich in EPA, or eicosapentaenoic acid. The problem with this particular strain is that their cell walls are extremely tough to break. So they're bilayered with an outer layer that's made of alginine. Alginine is some sort of cross-linking biopolymer, and an inner layer that's made from cellulose, with a little bit of protein embedment within that layer itself. So these cell walls are very rigid, and they're highly resistant to cell disruption by both mechanical and chemical process. And what my research in the past few years have found is that, and that I'm planning to continue as I build my group here, is that we are actually able to force nanochloropsis to somehow selectively decompose its cell wall without decomposing its omega-3 lipid if we incubate the nanochloropsis cells, or if we subject the nanochloropsis cells to an incubation treatment correctly under certain conditions. And what this treatment does, or what the selective cell wall decomposition does, is that it then helps or improve the yield of subsequent biomass processing. So in this particular case, you have your microalgae culture that's normally quite dilute, so that's about 0.1 weight percent solid. And you dewater, you remove the water, you get the slurry, which is generally about 11 to 20% solid. And what we did here is that we then pack this slurry into a tightly packed container, incubated in darkness at elevated temperature. And this simple treatment actually lead to the cells activated its anaerobic metabolism. So what happened here is, because you incubate it in darkness, the cells cannot photosynthesize, so they cannot replenish their oxygen. And because they're so tightly packed and they're metabolically active at 37 degrees, they start actually depleting oxygen from the slurry, and the whole system become anoxic. So you force the cell to activate anaerobic mechanism or anaerobic metabolism. And I won't go through the metabolic circuitry of the fermentation process, but essentially when they activate their anaerobic metabolism, they consume the sugar within their own cells or within their own biomass. And some of the sugar they consume are the structural sugar in the cell wall. So in the case of nanochloropsis, they actually consume the cellulose. So this is the nanochloropsis cell wall here. It's a TM image. And that's the cellulose layer. And then there's the outer alginine layer illustrated as well. So it's a bilayered cell wall, as I mentioned before. And after the incubation, you will notice that the inner cellulose layer has essentially been much more depleted and much thinner compared to the cell wall before the incubation. Now, of course, the incubation is not very, it's not kind to the cells. So I think a lot of them lose viability. Well, we're almost certain a lot of them lose viability after being incubated. And not that it matters, because you're processing it. You're not actually recovering this for growth afterwards. You're essentially disrupting it and extracting the lipid, or extracting the products from the cells after. So as I mentioned, as part of the anaerobic metabolism, nanochloropsis cells consume sugar in the cell walls, making the cell walls thinner. And as you see here, there's almost, I guess, a 75% drop in the cell wall thickness. And cells with thinner cell walls were easier to rupture. That's sort of, I guess, basic mechanics. I wouldn't say basic, because it's actually not that easy to mathematically prove it. But the idea is that if you think about it, you have a sphere with a particular wall, and if the wall is thinner, then it's actually easier to pop open. So this is sort of just an illustration here. You on both, at the top here, you have the untreated cells and the incubated cells, just sort of microscopic image. And then when you subject them to cell disruption, you will notice that there are more, with the untreated cell, most of the cells, a lot of the cells actually remain intact. So if you quantify the loss in the number of cells, you would say about 30% of the cells were disrupted or broken. But then with the incubated cells, you get a much more dramatic decrease in the number of intact cells. So in our case here, we are probably rupturing about 60 or 70% of the cells from the incubated slurry. And the more prolific cell rupture for the incubated slurry ultimately leads to a better mass transfer during the product recovery step, or the lipid extraction step in this particular case, and leads to a significant improvement in the lipid yield of the downstream process. So I guess my vision for the group is, first is to work on the upstream part, which is the cultivation with dairy processing waste, as well as waste from biogas production, but also to continue my research in the downstream processing, or in the biorefinery step, and that's to understand cell wall decomposition further and how it can be exploited for the biomass processing. So understanding, I guess, the biological mechanism of the cell wall decomposition. So that's something that we don't quite understand yet at the moment. I mean, I know that we noticed some conserved homologs of certain genes that are known to actually break down cellulose. I just want to say that we haven't actually, we've reported these findings and we've shown the physiological effects, but we haven't actually worked out the molecular mechanism underlying the actual cell wall degradation itself. And the idea is that we will try to do that. So understanding the process of cellulolysis, and then the glycolysis, and then the fermentation process that breaks down the cellulose ultimately into organic acids. And then my second sort of idea for the project is to develop a biorefinery system that's centered around this incubation step, integrating it somewhere in that value chain to help enhance the processing of the microalgae biomass. So combining it with different mechanical cell disruption process, as well as solvent extraction, or different types of solvents. And as I mentioned before, we've only applied to Nanochloropsis, so we would be keen to sort of expand that research and apply it to the other species as well. And that brings me to the end of my presentation. Thank you for listening, and I look forward to any questions, and I look forward to collaborating with colleagues here at this institute in any part of that value chain, whether it's cultivation, downstream processing, molecular biology, or even actually converting the products into applications. Thank you.